My PhD is the influence of climate change and plastic pollution on macrophyte detrital dynamics. So to introduce my PhD, there are three main topics, and that is plastics, climate change, and detritus. First, I'll get into plastics. So the plastics were first invented in 1909 by a chemist named Leo Baekeland. And between 1909 and the 1950s, it didn't really take off. It wasn't until past the 1950s that it started to exponentially be produced. And along with that was the exponential rise in plastic pollution around the world. It's gotten to the point where every year about 12.4 million tons of plastics enter the world's oceans. So where does it come from? Well, there are three main sources of plastic pollution in the world, and this is due to direct dumping, which is the direct input of plastics into the oceans, and this is usually in areas of poor waste management. The second is runoff, and that is when terrestrial plastics end up getting washed into these waterways and rivers, and then they're swept out into the oceans. The third way is through discarding from ocean-going vessels, and this is shipping, cruises, fishing vessels, and other such things that directly throw their trash overboard. So my literature review that there are three main areas of interest in which these plastics end up, and this is the sea surface, the ocean floor, and beaches. So plastics end up on the sea surface because they're lightweight and low density, and when they're on the sea surface, they're exposed to a bunch of environmental factors, and these environmental factors end up wearing them down, and things like biofouling and waterlogging end up making them sink. And when plastics sink, they end up on the ocean floor. And in the ocean floor, they end up being swept into areas of organic matter accumulations because of currents and other underwater waves. And both sea surface and ocean floor plastics can actually be swept up onto beaches by tidal processes, waves, and currents. And when they end up on these beaches in these lines of detritus, that is dead kelp and such, you can see it when you go on the beaches that in these lines of dead stuff, you see a bunch of plastics. And this has instigated things like Clean Up Australia Day to occur. Now, I want to get on to climate change. So since the 1850s, that's the start of the Industrial Revolution, CO2 levels have risen by around 40%. And coinciding with this increase in CO2 levels has been a temperature increase of 1 degree Celsius over the last century. And this is projected to rise in the future. Now, this is due to greenhouse gases such as CO2, carbon dioxide, methane, and nitrous oxide. And these greenhouse gases have absorptive and reflective qualities. And as we can see here on the right, solar radiation that enters the Earth's atmosphere usually bounces out normally. However, with these greenhouse gases that form in the lower atmosphere, the absorptive and reflective qualities make it so that it actually bounces back into the Earth. This means that radiation from the sun, as in heat, gets trapped in the Earth's lower atmosphere, which warms the globe. And this warming ends up warming the oceans as well because of the ocean's heat capacity. That is, that they absorb around 50% of the heat in the Earth's atmosphere. Now, this can have a slew of effects, such as phase shifts, where ecosystems change from one form to another. And this can be seen in Tasmania, where the distribution of an urchin called Cetrostephanus regersii has actually been extended down into Tasmanian waters. And when they're down there, they end up inhabiting these kelp forests, where they start eating the kelp and creating these barrens. And in these barrens, there's no kelp and therefore no productivity, meaning that there's little life going on there. Ocean warming also can cause an increase in algal blooms, usually due to the effects that warming has on the metabolic efficiency of organisms. This is due to organisms mostly being ectothermic in the ocean. That is, that they rely on the ambient temperature to help boost their metabolism. We also know that ocean warming increases the rate of decomposition of detritus, but we'll get into that later. Ocean acidification, on the other hand, is caused by high amounts of CO2 emissions in the atmosphere that end up dissolving into the ocean when it interacts with the ocean surface. And this enriches the oceans with more CO2 and lowers the pHs. And this can cause corals to bleach as they aren't tolerant to these new low levels of pH, and it causes them to die. Calcified organisms, on the other hand, like the snail in the right here, when exposed to ocean acidification, it actually erodes their shells. However, it's not all doom and gloom, as algae can actually benefit from this excess CO2 as they use it in photosynthesis. So what is detritus? Well, detritus is just the word given to dead organic matter. So that is just dead organisms. Now, this can take the form of coarse organic matter, which is usually large amounts of the whole organism. And then there's particulate organic matter, which is the coarse organic matter that's broken down into smaller pieces. And then when that particulate organic matter is broken down into microscopic pieces, it's usually called dissolved organic matter, which is usually the chemical components of this detritus. In the right here, we can see a mix of coarse organic matter and particulate organic matter. It's important to note that macrophyte detritus is highly productive, as macrophytes are photosynthetic and throughout their lifetime accumulate a lot of energy in the form of carbon and nitrogen within their tissue. This is important for when they break down and get consumed, it provides energy to other organisms. These mainly come from macroalgal forests, like we see up in the top right here, and seagrass meadows. Usually this detritus ends up on the seafloor or on beaches. As we see here in the bottom right, that's a lot of kelp detritus that has washed up on this beach. And you can see that the color has changed in the water here, and that's usually due to this dissolved organic matter. And due to this, detritus plays an important role in ecosystems. So why is detritus important? Well, it provides unproductive environments with energy and nutrient enrichment. So places like beaches, which generally have little to no productivity whatsoever in the form of primary productivity or secondary productivity, actually get a bunch of energy and nutrients from when kelp washes up onto the shores, providing them with a bunch of new energy. This usually enriches both epifauna, infauna, and other organisms that live on the beach. Detritus also underpins many food chains through what is called the microbial loop. So as we see in this diagram here, we have particulate organic matter that is absorbed by bacteria and consumed by other microorganisms, and they end up getting eaten by microzooplankton, which get eaten by larger zooplankton, and those zooplankton usually get eaten by fish, and those fish are getting eaten by bigger fish until we end up to top order predators. So this essentially means that they provide a base for many food chains. The energy that enters through detritus pathways can actually exceed that of herbivory. That is, when detritus is broken down, it actually provides more energy to the environment than, say, when a fish comes along and chews on some kelp. Now, buried detritus in the form of organic matter contributes to carbon burial, and that means that it alleviates the carbon contents of the ocean, which alleviates, say, the ocean acidification effect. Now, they have an interactive effect where, as you can see in this graph this series of graphs here on the right, that on the left we have seagrass and on the right we have kelps. Now, on the y-axis, we have biomass in the form of grams. And on the x-axis, we have the days in the number in days. And these top graphs show the effect of high, low, and no amounts of plastic have on the decomposition of seagrass and kelp. Here we see that high amounts of plastics significantly slow down the detritus decomposition effect, whilst low and the control are normal. Now, temperature, on the other hand, has the exact opposite effect. So here we see 23 degrees and 26 degrees, which is the temperatures these detritus were exposed to over this 40-day period. And here we see that the 26 degree, which is the ocean warming scenario, actually has an increased rate of decomposition. And this follows between the seagrass and the kelp. However, ocean acidification had no effect on either seagrass nor kelp. So for the purpose of my PhD, I will not be adding that into my experiments. So the objectives of my thesis are to evaluate the influence that various plastics have on the decomposition rate of dominant macrophyte detritus, and to identify the effect of ocean warming on detrital decomposition in the process of several plastic types, and to assess the effectiveness of drone technology via aerial drone surveys to calculate the deposition of RAC and its use as a monitoring tool. To achieve these aims, my thesis will cover the following 4 research chapters. Chapter 2 is the effectiveness of drone surveys in calculating beach RAC deposition in comparison to field surveys. So currently, to calculate the amount of biomass that's been washing up onto these beaches in the form of RAC is quite labor intensive and requires transect surveys and quadrat surveys, which can often lead to extrapolation, that uses extrapolation and can lead to overestimation or underestimation of the amount of volume that's washed up on these beaches. And this can lead to poor management decisions when it comes to removing this RAC off of the beach, which can actually reduce productivity if it's poorly mismanaged. So the objectives of this project is to evaluate the effectiveness of drone survey techniques for identifying the size and mass of beach RAC. I want to compare the accuracy and precision of drone surveys to on-the-ground field surveys in measuring the size and mass of beach RAC. I want to develop a technique that can appropriately estimate the biomass and aerial extent of beach RAC with drone surveys, evaluate the effectiveness of drone camera resolution to pick up on plastics mixed with macrophyte RAC, and measure the amount of beach RAC at several beaches in the mid-north coast of New South Wales, Australia. So the methodology will require the use of 2 drones, Phantom 4 and a Mavic 210. This is just to see if either one has is more effective at field surveys. We'll be using 3 different altitudes, 60, 80, and 100 meters. And we'll be assessing whether or not the zoom function is useful for these surveys. Now we want to calculate the RAC volume using drones. So by doing this, we'll get the RAC volume that we know of, so no, low, and high, and we'll put them out there and use the drones to fly over and see if they can calculate the volume based on what they see. I also want to try and see if drones can be useful in the identification of plastics mixed with this RAC by using different plastic types, plastic sizes, and the amount of plastic within a square meter. Chapter 3 is on measuring the decay of buried macrophyte RAC under the influence of plastic debris. So beach RAC is often buried by sand, and this is through wind and wave action that ends up pushing the sand over this RAC and burying them. However, plastics often tend to accrue with these beach RAC. Therefore, it's important to try and find out what effect these plastics may have with this rack when it's buried. So with this study, I want to measure the decay rate of macrophyte rack buried in differing depths of sand. I want to observe the influence of differing concentrations of plastics mixed with buried macrophyte rack. And I want to identify any differences in infaunal communities in buried rack with and without the presence of plastics. So the methodology for this will require about a 100-meter line where we'll have random points that will dig 10 by 10 centimeter squares at two different depths of 20 millimeters and 50 millimeters, where we'll place these litter bags filled with detritus in them and bury them and place a stake next to them for collection purposes. They'll be collected at intervals of 5, 10, 20, and 40 and 80 days. And each litter bag will contain one of the three following plastic treatments of none, 0.1 grams as our low treatment, and 0.8 grams as our high treatment. The samples that we retrieve from the litter bags will be dried and weighed so we can get an idea of the decomposition rate. And once the litter bags are removed from the sand, we want to take sediment cores from underneath them to see if there's any difference in infaunal abundance within this sand underneath certain treatments of litter bags. Chapter 4 is the effects of various common plastic types on the decomposition of sargassum in a warming ocean. So as we know, plastics often accumulate in detritus depositions. And we already know from earlier that high amounts of plastics actually slow down the decomposition rate of both seagrass and kelp. So what I want to do is, rather than looking at one type of plastic, I want to look at various types of plastics. And the aims of this study are to understand whether there is a difference in the decomposition of macroalgae under the influence of various plastic types, to determine whether the ocean temperature influences the decomposition of macroalgae under these various plastic types, and observe if the carbon-nitrogen ratio of the detritus of sargassum tissue changes throughout the decomposition process under various plastics. The methodology for this will be using a mesocosm setup, and that includes 20 header tanks that will be feeding water into a series of 20 raceways underneath them. And there'll be two temperature treatments for this. There'll be current temperatures, which is around 23 degrees, and future temperatures predicted by the Intergovernmental Panel on Climate Change, which is around 26 degrees. There'll be 4 intervals for collection, 5, 10, 20, and 40 days, so we can see how the decomposition rate goes. There'll be 5 various plastic types as our treatments, and that is low-density polyethylene, the old consumer shopping bags, high-density polyethylene, the new bags that have been introduced by places like Coles and Woolworths, biodegradable starch bags to see if biodegradable bags have any influence at all, PVC as polyvinyl chloride. Because polyvinyl chloride has been shown to be quite toxic to microorganisms, it'll be interesting to see how these affect the decomposition of detritus. And lastly is polyethylene terephthalate, which is your plastic bottles, which have been found all over the world in the oceans. Chapter 5 will be the effects of plastic fishing gear on the decomposition of sargassum in a warming ocean. So netting from commercial fisheries has been found throughout the world as pollution in the oceans. The same goes for recreational fishing gear that's usually cut off by fishermen who end up getting snagged on logs or whatever. And here you can see on the right that this person has found a bunch of lures and tangled line in just one area. So this study aims to evaluate the influence of polyamide netting as commercial fishing gear and soft plastic lures as recreational fishing gear on the decomposition of macrophyte detritus. I'd like to determine the effects of netting and lures on detritus decomposition within an ocean warming scenario, and identify whether the nutrient composition as carbon and nitrogen of macrophyte detritus changes throughout the experiment under influence of netting and lures. Now, the methodology for this experiment is the exact same as Chapter 4, except we will have more intervals for collection to see how the decomposition is in the early stages. Now on the timeline of my PhD thesis, we are here in November and December, and I have said that chapter 4 will be done by the end of December, and I am on track to finish that as the experiment has already been finished, and I'm going through writing it up and doing the appropriate analyses. I'm already getting ready for chapter 5 and organizing experiments, so I'm already on time for that as well. Since chapters 2 and 3 are field related and dependent on weather, I've pushed them to the back so I can have some more time and leniency just in case anything comes up that inhibits me from going out and doing the field surveys or anything else that may inhibit me. I needed 2 permits for collection of macrophytes in the Solitary Islands Marine Park, and these are these 2, and just allows me to collect the macrophytes that I need for my experiments. I'll also need to get my sub 7 kilogram drone remote pilot license so I can do my field surveys for chapter 2. So in conclusion, plastics are detrimental to ecosystems and climate change will have an adverse effect on the marine environment. Detritus is important for ecosystem functioning and food webs, and detritus is affected by both plastics and climate change. Due to these things happening in tandem, it's important to assess what impacts they may have together. My thesis aims on providing a broader set of knowledge on how macrophyte detritus is affected by pollution and climate change, as well as allows for effective management strategies to be developed with proper understanding of how detritus is impacted by this pollution and ocean warming. Thank you.